presentation on the heart, and I want to go here to have some material to work from. I'm not going to go through this in a lot of detail. I'll skip over some things just to mention they're there, and you need to spend a little bit more time with that on your own. The point of sort of the lab material for today is to, one, look at the heart anatomy, which is what I'm going to concentrate here in the first half, which will essentially culminate with you looking at the heart models that I have out. I was hoping to try something out today, which was I have a camera that I was going to plug into the computer, and I could switch from capturing the screen to using the camera. And I was going to have the camera on the model, so when I'm showing you stuff on the model, I'd capture it for the presentation also. I won't be doing that, which you don't care about because you're here and you're going to see it anyways. But for those of you that are listening to this online, there's going to be this weird pause as I'm not recording what I'm showing everybody. What I was hoping to do was to plug the camera into the USB on the keyboard here, but the camera needs more power than that USB port can provide. And the cord on the camera is too short for me to plug it in there and actually use it because it get that high. I do have a USB extender at home, so I'm going to use that in the future for when I'm talking about models, which I don't know if that's going to actually work, but I want to try it out. But for today, for you here in the room, I'll be showing you the models, and it just won't be on what's captured for the lecture. Now, I'm not going to go through this stuff in a lot of detail. This is just sort of talking about where the heart is located. It's obviously within the thoracic cavity. The space in the thoracic cavity where it's found is specifically called the mediastinum, which is not just where the heart is. It's just the space between the two lungs, which contains a number of different organs. And these pictures are just getting at what we see there. So at the top, there's the heart. And so the mediastinum is the area between the two lungs. And then this cross-section through the thoracic cavity, you can see the position of the heart relative to the lungs and the other structures that we would find within the mediastinum there. The next couple of pictures down here are getting at the tissue that surrounds the heart, what we call the pericardium. And we're going to see this sort of thing a bit in this course. We're going to meet it here for the heart, and then when we get to the respiratory system, we're going to see the same thing in relation to the lungs, and then in the digestive system, we're going to see the same thing in relation to the stomach and intestines and other digestive organs in that area. What's true across all three systems there is what we're talking about is called a serous membrane. And a serous membrane, and you can go back to the tissues chapter where membranes are introduced, a serous membrane is a simple squamous epithelium and its associated areolar connective tissue. So in the picture up here, part of it that they're highlighting, this little inset, the serous membrane is really just the epithelium and the areolar tissue associated with it. That simple squamous epithelium of a serous membrane is often referred to as the mesothelium, which is referring to this epithelium in the middle of the body, within the body cavities. You've probably heard a reference to mesothelium before, but not that word exactly. You've probably seen lawyer commercials that are talking about class action lawsuits about mesothelioma. That's talking about specifically the mesothelium surrounding the lungs. And the class action lawsuit is an industrial lawsuit, I guess you'd call it, against employers that had people working for them that were exposed to asbestos, like mine workers, miners, coal miners or something like that, maybe. Actually, my next door neighbor, he doesn't necessarily have this problem, but both of his brothers had mesothelioma. I think he said they worked with an oil company, and part of what they did was cleaning out oil tanks or something like that. And so they were exposed to asbestos in that process. How, I don't know, but so this class action lawsuit is about cancer of the mesothelium, specifically around the lungs, because people would be breathing in asbestos. But mesothelium is really just referring to that epithelium that's part of a serous membrane. And in all of the situations that we're talking about, around the heart, around the lungs, and around the digestive organs, the membrane is sort of a two-layered situation, sort of. It's really one layer, but it wraps directly around the outside of the organ, what we call the visceral layer, in this case, the visceral pericardium. That's not labeled here. We'll see it in another picture. And then it extends away from the organ and lines the inside space of the cavity, which is the parietal layer. So up here we do see parietal pericardium. The word parietal is referring to the body wall, essentially. So it's the layer that's lining the inside of the cavity. And then visceral means having to do with an organ. So it's the layer that's directly surrounding the organ. It is actually one continuous membrane that sort of folds over right here. The apical surface of the membrane on the visceral side is facing the apical surface of the membrane on the parietal side. And between those two is a little bit of space. That space is filled with fluid. The point of the pericardium, or one of the major points of the pericardium, is for lubrication. So as the heart's beating within your chest, the pericardium surrounding it, and that fluid within the pericardium cuts down on the friction of the heart moving against what's surrounding it. Then they're showing you the parietal pericardium here, which I pointed out a second ago. But really, there's more to what's in this inset. There's some fibrous layer on the basal side of the membrane. Here they refer to that as the fibrous tissue of the pericardial sac. You might just as easily hear fibrous pericardium. That's dense, irregular connective tissue that sort of surrounds everything and anchors it in place. Down here at the bottom, we can see how that fibrous tissue is connecting it down. Not in the picture, but just below this would be the diaphragm, so it's anchoring it to the diaphragm. And elsewhere, it's anchoring it in place to the rest of the walls of the mediastinum. I'll come back to this picture in a second because there's a bit more that I want to talk about, but the next picture sort of goes a little bit further. If you're having trouble visualizing what this membrane is, this picture shows a balloon that's like mostly deflated. And if you put your fist into that balloon, and your fist is about the size of the heart, the balloon will wrap around your fist, and there will be some of the wall of the balloon that's actually touching your skin, and then it folds over and part of it that's on the outside. And that represents the two layers. So the inner versus outer walls here correspond to the visceral pericardium and the parietal pericardium. Now up here, they don't call it visceral pericardium, they call it the epicardium, which is another name for that, which has to do with structure of the heart, which we'll see in a sec. But if this helps you kind of visualize what's going on here, that's helpful. If not, I've always thought this is kind of a silly picture. One thing to point out here, they label the space in between, they say that airspace. Let's pretend this is a water balloon because it's going to be a little bit more accurate. So instead of airspace, that's where the little bit of water would be in this rather sad little water balloon. And that would represent the serous fluid that's in there that helps to lubricate the heart as it's beating. So again, if this visualization helps you understand a little bit, that's great. But if not, don't worry about it. What I want to go back to this picture for real quick is a little bit more about the structure of the heart. Down here, it's pointing out what's called the apex of the heart, which is kind of the pointy bottom part of the heart. And then this line up here is showing the base of the heart. The heart is pretty much on midline, but it's a little bit off to the left side. And where it points off to the left side, sort of comes to a point right there, that's the apex. And then the other side is the base, which is where it's connected to everything. The words apex and base, if we're using those words to describe a mountain, would be talking about the bottom or base of the mountain and the top or apex of the mountain. Here, the base is the top of the heart and the apex is the bottom of the heart. And really, apex is used because it's the pointy end. So the pointy end of the mountain is the apex of the mountain. So that's where that comes from. So I just want to point that out. Actually, in this picture with the balloon I was just showing you, they suggest the wrist would be like the base of the heart there. And that shows you the pericardium is wrapping around all the heart except for the base, which is where it's connected to everything else. So to talk about that term epicardium for a second, here we see the parietal pericardium with the fibrous layer out here. And then the visceral pericardium is the layer of mesothelium and its connective tissue next to it, which is also considered the outer layer of the heart as an organ, which we call the epicardium and also called the visceral pericardium. As we move in from that, the next layer, which is the majority of the wall of the heart, is the myocardium, which is the cardiac muscle tissue, and associated connective tissue, which helps to hold it all together. And then the innermost layer is the endocardium. That is also a simple squamous epithelium and its associated areolar connective tissue. It just lines the inside surface of the heart, making it a continuous, smooth surface. That is then continuous with the simple squamous epithelium that's the inner lining of the blood vessels. Here we call that the endocardium. When we get to the blood vessels, we call that the endothelium. But really, it's just one continuous layer of simple squamous epithelium so that the cardiovascular circulation is one continuous path throughout the body. Up in the top here, they show us the orientation of muscle fibers within the myocardium. The muscle fibers are wrapped around the chambers of the heart, which we'll talk about a little bit. And for the bottom chambers, it wraps around, kind of tucks in, the way they've drawn it, at the apex. Okay. So the fibers, the direction of contraction, are kind of oriented very specifically around the three-dimensional structure of the heart. I was going to go to, apparently I don't have the picture I thought I had in here. So actually from this, I want to switch to the book where there's a good picture of this. And I should have opened this up. So having just pointed out the layers of the heart, I want to talk a little bit more about the tissue that makes up the myocardium. So anyway, since I'm in the book, this first part is sort of talking about its location. And then in the next part, it's talking about the electrical activity of the muscle. And we'll get to some of that in a little bit. But in keeping with talking about the tissues in the heart, I want to show you this picture, which details what cardiac muscle looks like. The micrograph that's here is a little small to really make out everything that's going on. I would suggest that you might go to do an image search and look up cardiac muscle tissue, and there's lots of micrographs that show what this is pointing out, perhaps a little bit better. One thing that's not evident in this micrograph, and actually not really that evident in too many of the micrographs you'll find online, cardiac muscle tissue is striated. So we should be able to see striations across the fibers just like we do with skeletal muscle. The striations are a bit more subtle. They're a little harder to see, and that's why we don't usually see them. I've had pictures out of textbooks that do have the striations pretty well represented. In the cardiac muscle slides that we have here, which you're welcome to look at on your own at some point, the ones that are kind of overstained, we can see the striations, but it's really kind of hard to see in plainly stained tissue. But down here in the drawing below, it shows you where those striations would be. Of course, in A&P1, you learn those striations in skeletal muscle are really just the visibility of the alternating light and dark bands in the sarcomeres, the thick filaments and where there's no thick filaments found. Okay, and so the fact that cardiac muscle is striated just like skeletal muscle is really telling us that contraction is based on the exact same model that we learned in A&P1, which is the sarcomere sliding filament model thing. If you're not familiar with that, please be sure you go back to the muscle tissue chapter, which is chapter, let me think about this for a second. 1, 2, 3, 4. I think chapter 5 in unit 2 in the book. So you might want to go back there and it'll remind you how all of that works. Part of the reason why the striations are a little bit more subtle in cardiac tissue is because the myofibrils, which are made up of the sarcomeres, are not so densely packed within the cell. There's a bit of space between some of them. There are mitochondria represented here, which we'd also see in the skeletal muscle, but the space between them is a little bit greater. So they don't really stain as darkly because they're really not as much a part of the tissue. Now, the other thing, and what we can see in this picture, especially because they're pointed out, there are these lines that go across the tissue, and you could certainly find better representations of that through an image search online. Those are what are called intercalated discs. They are basically where the cells come together. And in the drawing up here, the intercalated discs are represented here. Now, in the micrograph, what you're seeing is a heavy pattern of staining right at that line. You don't see this sort of zigzag kind of picture line in the picture micrograph, but that represents the cell membrane there. The cell membrane in one cardiac muscle cell kind of looks like egg crate pattern, and then the cell membrane that kind of fits into it is going to be the opposing pattern, so they kind of interlock. What makes it stain darkly in the tissue is that there are a bunch of proteins there holding the cells together, which is represented here. There are attachment points called desmosomes, which are common types of attachment points. They're introduced in the book originally in the tissue chapter, where it's talking about how epithelial cells are attached to each other, and they use desmosomes also. So these are just cell adhesion points. There's also going to be some proteins where the myofibrils come to the edge of the cell and they kind of attach. And notice that the myofibrils in one cell kind of line up with the myofibrils in the next cell, so that all of the contraction kind of happens in a very linear fashion across the cells. Where the cell membranes come together kind of weave in and out like this that we see, and that corresponds to the zigzag kind of picture here. But then it turns and it runs along adjacent to or parallel to the myofibrils that we see for a little while, and the proteins that we find in there are a little bit different. These are what are called gap junctions, and gap junctions are just shared ion channels. So again, if I had the camera set up, I'd be putting this on the screen too, but let me just describe it for everybody's imagination that's listening to this. I have little pieces of tarp here. And if you buy a tarp at the store or whatever, you usually have a little piece on the outside of the package so you can see how thick the tarp is. And for some reason, they usually put those in with a little grommet in, because the tarp's always going to have little points where you can tie the tarp down. And that's where these came from. So I have two pieces of tarp here, each with their own grommet, and the grommets represent ion channels so that ions could pass through a membrane. For a gap junction, what it is the cell membrane of one muscle cell and the cell membrane of another muscle cell line up, and their individual channels match up and link together. So it's a sort of a double channel that allows ions to move from one cell directly into the next cell. So if this cell at the bottom is depolarized and it's got a higher concentration of sodium than normal, then that sodium is going to move through the gap junctions into the adjacent cell and help to depolarize that cell. Now, the gap junctions kind of tell us something about how the heart does something, solves a problem that's very important. And this problem has to be solved by skeletal muscle and cardiac muscle. They just solve it in different ways because those two types of muscles are different in a certain aspect. What they both have to do is to coordinate the electrical activity of the tissue so the contraction all happens at once. You don't want just a little itty-bitty microscopic cell contracting by itself and all of its neighbors being off by, milliseconds or something like that. But if you don't coordinate the contraction together, you're not going to get the muscle as an organ working properly. What skeletal muscle does is it fuses all of the little cells together. So a muscle fiber in a skeletal muscle runs pretty much the whole length of the muscle. There are a bunch of nuclei in the cells because all of the little myoblasts from development fuse together to make the one huge supercell that is the myofiber in the mature tissue. And it goes from the origin to the insertion of the muscle. So when that muscle depolarizes, when that fiber depolarizes, the whole fiber contracts. And the whole thing leads to moving the insertion towards the origin. Skeletal muscles, however, are very linear, so they can have one long cell going the whole distance. Cardiac muscle is different. The heart is a three-dimensional structure. It has to have that three-dimensional shape. And you can't have one cell wrapping all the way around, kind of like that picture I showed you a little bit ago. Instead, you have to have individual cells that each have their own individual structure, providing the three-dimensionality to the tissue. But you want to have them all coordinated and contracting together. So what the cardiac muscle tissue does is the cells are anatomically separate. So this is one cell, and we see it's one nucleus here. The inset shows the intercalated disc between this cell and this cell, which is one complete cell. It has its own nucleus, et cetera, et cetera. Here's another cell out here. We can't see its nucleus because I haven't cut it open, but this is another cell, and then there's another cell here. At each of the intercalated discs, there are gap junctions so that the cells are electrically coupled together. When one cell depolarizes, its neighbors are going to automatically depolarize. And so just like depolarization spreads along the length of the one skeletal muscle fiber so that the whole thing contracts, depolarization will spread from one cell to the next, to the next, to the next, so that they all contract together. And that's happening because of the gap junctions here. Question?
Got a picture missing the sarcoplasmic reticulum.
Yeah, they've simplified the picture, certainly. But that's not crucial to understanding what we're seeing here. The intercalated disc is what we see under the microscope. We don't see organelles like sarcoplasmic reticulum and that sort of thing. We don't see mitochondria either, but they can kind of leave those out because we don't really see it at this level. We don't see the striations, but it is striated tissue, and when it's stained in a certain way, you can actually see them. So we would see the banding pattern of the sarcomeres that are drawn in there, certainly. But yeah, all of that stuff, the sarcoplasmic reticulum, et cetera, that you learned about in skeletal muscle, that all applies to cardiac muscle too. So like I said, I wanted to go to the book to show you this picture because for whatever reason, I don't have it in this presentation, but that would be the obvious thing to do here after talking about the tissues. And in this drawing, actually, you can see they've drawn in the intercalated discs. So this is one cell here, this is another cell, et cetera, et cetera. But they're all electrically coupled together. So when depolarization starts in one cell, it just spreads through all of the other cells because they're all interconnected there. Oh, wrong direction. So now going over to this part. This is getting at the more gross anatomy of the heart. And I want to address this stuff. This is going to lead up to looking at the models here. And depending on exactly where I am time-wise with this, I might have us take our break, and in that break, which will be longer than usual, I'll also have you take some time to look at the models. And I'll explain what I want you to get out of that when we get to there. But let me use the presentation here to kind of explain where things are. Also, I'm going to be switching back and forth from the presentation to this blank page because I also want to add in some things here. So what we're going to talk about here as far as what the anatomy of the heart is about is that there are 4 chambers, there are 4 major vessels, and there are 4 valves. And I want to list these out for us. Whoops. As we talk about them. This list that I'm putting together here is kind of getting at, it's better to kind of consider it in the exposed view, but this picture has a bunch of labels on it, a little too much information. So with this list, I just want to provide you with the basic information I want you to get out of this. And this is the stuff that I want you to look at on the models. So first off, there are 4 chambers. They are 2 on the right, 2 on the left, 2 superior and 2 inferior. The superior ones, let me go back to this picture here, are called atria, atrium being singular. And then the inferior ones are called ventricles. So putting the right and left and atrium and ventricle stuff together, we have the right atrium, the right ventricle, the left atrium, and the left ventricle. Now, I'm specifically listing them in this order, and I'm going to kind of do the carry through with this with the vessels at least, because we also want to consider the movement of blood, how it's pumped through the heart. And the right and left sides actually are kind of independent pumps. They work together, which is an efficient use of the muscle tissue, but they're actually separated. Right down the middle, oh, sorry, question?
Yeah, real quick, which ones were superior? Which ones were
the atria are superior, the ventricles are inferior. So right down the middle, separating the right from the left half, is this continuous wall of tissue called the septum. The word septum is just a general word for a wall. You have a septum in your nasal cavity, or separating your right and left nasal cavities. A deviated septum is referring to the nasal septum, not to the cardiac septum. So the 2 sides are separate. The blood moving through the right side doesn't mix with the blood moving through the left side. In a perfect world. Sometimes there are holes between the septa, between the two sides, called septal defects, the most common of which is an atrial septal defect. In the fetal heart, since the lungs are not functioning in a fetus, the right and left atria are actually connected. There's a hole in the atrial septum, which doesn't always close up. So some newborns will have an atrial septal defect, which for most people will just finish growing and close up eventually shortly after birth. But for some people, the atrial septal defect remains and the right and left sides mix, which is not supposed to happen. So the right side receives used blood, deoxygenated blood, from the body into the right atrium. And then it moves from the right atrium into the right ventricle, and the right ventricle pumps the deoxygenated blood to the lungs. And then lungs oxygenate the blood, and it comes back to the left side of the heart at the left atrium, and then from the left atrium it goes to the left ventricle, where it's pumped out to the body. So the right side of the heart is really pumping the deoxygenated blood from the body to the lungs, and the left side is pumping the oxygenated blood from the lungs out to the body. And to move the blood in and out of the heart, we need to work with vessels. The blood from the body coming into the right atrium comes in through the vena cava or vena cava. There's an inferior and a superior vena cava. This inferior vena cava is carrying blood from the lower body up to the heart. The superior vena cava is bringing blood from the upper body down to the heart. So it's basically at the heart, everything above the heart comes in from superior, everything below the heart comes in from inferior. Then the blood goes from the right atrium to the right ventricle, and it's pumped from the right ventricle to the lungs through the pulmonary trunk, which is the trunk of the pulmonary artery, or arteries. A single vessel emerges from the right side, and then it branches into the left and right pulmonary arteries. I should say to the arteries. And then coming back from the lungs, the blood travels to the left atrium through the pulmonary veins, and the pulmonary veins are connected directly to the left atrium. And then the blood goes from the atrium to the ventricle, where it's then pumped out through the aorta, which is an artery. So let me jump over to the picture real quick and point these things out. So again, the two upper chambers are the atria, the one on the right and the one on the left. And then the two lower are the ventricles, the one on the right and the one on the left. Blood from the lower body comes up through the inferior vena cava, and from the upper body through the superior vena cava, both of which drain into the right atrium. And then the right atrium pumps the blood into the right ventricle, and we'll talk about the valve between them in a second. And then the ventricle pumps that blood out into the pulmonary trunk, which we see right here. That then splits into the pulmonary arteries, one to the right and one to the left. And then here we see the left side's already branching further, going to the lungs on their respective sides. The blood comes back from the lungs through the pulmonary veins. There are two coming directly in from the left lung to the left atrium. There are two coming in from the right lung, which are not visible in this picture, but they're going to attach to the left atrium on the posterior side of the heart. Okay? And then the blood's pumped from the left atrium into the left ventricle, and the left ventricle pumps that blood out to the aorta, which we see arching over here. Now, you'll notice that some of these are colored blue and some of them are colored red. A lot of people would like to think blue is for vein and red is for artery. And the two that I'm pointing at here are, in fact, blue for vein and red for artery, because the vena cava is colored blue and the aorta is colored red. But those two are the vessels that are connecting the body to the heart. So used blood coming in through the vena cava is blue, and the fresh blood being pumped out through the aorta is red. The color is really about the oxygenation state. So deoxygenated blood is blue, oxygenated blood is red. And really, deoxygenated blood isn't really blue, it's just a very dark reddish purple kind of color. But blue is the standard color we use for that. The pulmonary artery and pulmonary veins are colored opposite of what you might expect. The pulmonary trunk and pulmonary arteries are blue. Again, they're pumping deoxygenated blood, or carrying deoxygenated blood to the lungs. And then the pulmonary veins are red because they're transporting the oxygenated blood back to the heart. Arteries are vessels that carry blood away from the heart, whether it's the aorta carrying it away to the body or the pulmonary arteries carrying it away to the lungs. You can remember artery and away both start with A. And that mnemonic helps you remember it. And so veins carry blood towards the heart. Okay, there's not a neat mnemonic for the V in vein, but that's the opposite. So the vena cava is a vein carrying deoxygenated blood to the right atrium. The pulmonary veins are carrying red oxygenated blood to the left atrium. Okay. So it's the artery vein thing is just about the direction of blood movement relative to the heart. Arteries away. And the color is really just about the state of the blood in that. Now, you can go on as far as this class is concerned with, if you're more comfortable thinking about it, that veins are blue and arteries are red, as long as you understand that that's the opposite for the pulmonary circulation. So the pulmonary arteries and the pulmonary veins are blue and red respectively instead of the other way around because of what they're carrying to and away from the heart, the lungs. We are not going to discuss the specific anatomy of the pulmonary circulation. We will talk about the systemic circulation, but you're not going to learn the names of any other arteries in the pulmonary circulation. So as far as we're concerned, if a blood vessel has pulmonary in its name, it's blue for artery and red for vein. Otherwise, everything else is red for artery, blue for vein. Okay. And we're going to be learning a number of other blood vessel names, but they're all going to be in the systemic circulation. Okay. So those are the chambers and the vessels. The valves, I'm going to present slightly differently. Because there's two types of valves, two of each giving us 4 valves altogether. One type of valve is called a arterioventricular valve, which we sometimes abbreviate AV, and the AV is for arterioventricular. I didn't say that right. Atrioventricular, sorry. Atrioventricular, there we go. So A for atrio, V for ventricular. And I'm putting the abbreviation before the word valve to point out that the V in AV is not for valve. So it's kind of a mouthful to say atrioventricular, and as I just demonstrated, it's very easy to mistake that A part of the word with other things there. So I will always refer to them as AV valves, which refers to atrioventricular. And the name comes from the fact that they are valves that separate an atrium from a ventricle. On the right side, that is called the tricuspid valve. And on the left side, that is the mitral valve, which is also known as the bicuspid valve, but we call it the mitral valve. Now, from that, it might seem like the tricuspid valve is important because it has 3 cusps, and the mitral valve is important because it has 2 cusps. But actually, all valves except for the mitral valve have 3 cusps. So calling the tricuspid valve isn't pointing out anything special about it. That's just the name that it has. Okay. The other type of valve is called a semilunar valve. And it's not terribly common to actually abbreviate this, but in keeping with what we just saw for AV valves, if you see an abbreviation, it's going to be SL for semilunar. But I never actually refer to them by that abbreviation. It's just a little awkward. On the right side, oh, I should have said this. Semilunar valves separate the ventricle from an artery. On the right side, that's called the pulmonary valve. And on the left side, that is the aortic valve. So what are the valves actually named after?
Direction, right?
No.
The direction of blood flow.
No. The semilunar valves are named after what? Which vessels? Well, yeah, specifically the pulmonary and the aorta, but the arteries that are on the far side of the valve. Okay. So just to include that. Now up here, I actually wrote these two lists out so they correspond. So right atrium is receiving blood from the vena cava. Right ventricle is pumping blood into the pulmonary trunk. The left atrium is receiving blood through the pulmonary veins. The left ventricle is pumping blood into the aorta. Down here, I don't quite do it that way because I have the right and left of the atrioventricular and the right and left of the semilunar. If I were to rearrange this to match what we have up above, the tricuspid valve is between the right atrium and the right ventricle. The pulmonary valve is between the right ventricle and the pulmonary trunk. The mitral valve is between the left atrium and the left ventricle, and the aortic valve is between the left ventricle and the aorta. Okay. So the order in the valves here doesn't match the way that I've ordered the chambers or vessels, just because I wanted to stress the AV versus semilunar valve classification. And that's important as we look at the picture here, because the AV valves have sort of a special aspect to them, which is they're attached to the ventricular wall. These little lines coming off of the edges of the cusps of the valve are called chordae tendineae, which is just Latin for tendinous cord, which is how I'll tend to refer to them. And so these little tendons attach the edge of the valve to muscles in the ventricular wall. And so we see that for the tricuspid valve over here and for the mitral valve over here. The semilunar valves, however, do not have tendinous cords. Now, this particular picture doesn't show them separated out like that. We don't see the opposite side, but that's the case. They're just these flaps of tissue that open and close. Now, actually, here is a dissected heart, and the tendinous cords, and the valves for that matter, are whitish in color. They're dense connective tissue, so they're mostly just collagen. And we can see the tendinous cords coming off of the tricuspid valve here and the mitral valve over here. And then we can see part of the aortic valve right here, and we don't see the pulmonary valve because this slice is taken posterior to that valve. So this is an anterior view of the heart, but it's been, so anterior meaning looking at it from the front like this, but the section's been taken through where the aorta attaches to it. So we've actually cut all of the front part of the heart off so that the pulmonary valve and where the pulmonary artery attaches isn't even in the picture. Okay. But the point here is we can see the aortic valve, and it does not have any little tendinous cords attached to it. Back to this picture. The picture is drawn in so we can see the pulmonary valve here. We don't see the aortic valve because the aortic valve is actually posterior to it. So as we're looking at this from an anterior view, we don't see that aortic valve. The similar picture in the book actually fudges the picture a little bit, and it has the aortic valve poking out here, but actually you wouldn't see that. So this picture that I'm using here is a little bit more anatomically correct, a little less student-friendly, I suppose, but that's fine. You're going to be looking at the models later on, and I want you to pay attention to how you can see the pulmonary valve very easily right here in front, and the aortic valve you can't see from an anterior view because it is obscured by where the pulmonary valve is. Okay, so sort of notice the arrangement in 3 dimensions on the model relative to what the pictures here are demonstrating. Now I want to talk a little bit about the function of valves because that's kind of the next part of what we see here after the dissected heart here. And so this is what I want to use to talk about the function of valves. This is obviously not a picture of the heart. This is a picture of a lobster trap. And this is an analogy I like to use to explain what valves are for. Okay, valves are kind of like little funnels. They make blood move in one direction. The lobster trap entry is kind of the same concept. So this is actually from a patent for a particular type of lobster trap. I don't remember exactly where I got it, but right here in the middle, there's this pocket, which is where you'd put the bait. Now, I've never bothered to try to capture lobsters. I don't really like lobster. But when I was a kid, I lived down on the Gulf Coast of Texas where we went crabbing quite a bit. So we had crab traps that are very much like this. And the bait we used for crabs was chicken. Okay. Why crabs would be interested in chicken, I have no idea, because a crab has never naturally met a chicken, I would imagine. But they put a little chicken in here. This lid here, of course, would be closed. But in the water, the chicken bait there is going to give off some molecules that dissolve in the water and are therefore, I suppose, attractive to the crab or the lobster or whatever you're trying to catch. And so they crawl towards the trap, and they can't get to the bait directly, so they crawl along the outside of the trap, and they get to one of the openings, and they crawl through. The opening is wider on the outside than on the inside. So the crab or lobster or whatever crawls in to get to the bait and eat. Then it can't crawl back out. Okay. I use this to talk about heart valves or valves in general because it's kind of the same idea. Imagine, for instance, the outside of the trap here is the atrium and the inside of the trap is the ventricle. That kind of funnel shape is making sure that the blood flows in that direction. Now, crabs or lobsters are solid things, and so getting out involves squeezing the lobster or crab through that little hole. Blood, of course, is more of a fluid tissue. And so if we just had a funnel, we could certainly get the blood to move back up through the funnel because liquids will do that. So really what happens in the valves is they open and close. So when they're open, they're kind of funnel shaped like this so that blood moves down through the valve. But when the ventricle is full, the blood will be filling up that space and pushing against the sides of the cusps, pushing the valve closed. Okay. And then when the valve's closed, the blood can't move back up. Now the pressure of the blood building up within the ventricle, as I'm describing it here, is going to keep the valve closed. But the atrioventricular valves have those tendinous cords because the pressure is a little overwhelming. The pressure when the ventricle is filling is very, very low, in the range of less than 10 millimeters of mercury of pressure. As the pressure builds up in the ventricle, as it's contracting, the pressure is going to go up into the, approaching 100 millimeters of mercury, and even higher when it's at full contraction. And so that much pressure pushing back against this valve, going from almost 0 to about 100, could easily blow the valve back the other direction. So the tendinous cords, kind of like my fingers, are on the edge of the valve, and so it's closed by the pressure pushing against the valve, but then the tendinous cords are attached to the ventricular wall, and when the ventricular wall contracts, it pulls on those tendinous cords, keeping the edges of the cusps down, and the blood pushing back against it can't blow it the other direction. Semilunar valves don't need that because the pressure differential across those valves is not as great as across AV valves, and they'll stay closed on their own pretty much fine. So, but that's my little analogy for how valves work. And then this is a picture kind of showing this. The picture is specifically of the bicuspid valve or the mitral valve. We tend to pay the most attention functionally to what's happening on the left side of the heart because that's the one that's pumping blood out to the body. But the right side works on the same principles. The big difference here is that we have 2 cusps in this valve, whereas the tricuspid valve would have 3 cusps. But we have the open valve with blood flowing from the left atrium into the left ventricle. And then once the left ventricle is full, that blood's going to be pushing back against the valve. But the tendinous cords, which are along the edges of the valve here, are being pulled tight when the ventricular wall is contracting. And that helps to keep it closed so it doesn't blow out the other direction. Okay. The pressure across the semilunar valves is not enough to need those cords to keep it closed like that. So. Now this picture, and there's a similar picture in the book, is just showing you the circulation through the heart relative to all of the blood vessels out in the body. If we kind of think about the direction that blood moves through the heart, the blood enters the right atrium from the vena cava, goes through the tricuspid valve into the right ventricle, and then through the pulmonary valve into the pulmonary trunk, and eventually to the lungs. And then from the lungs back to the left atrium through the pulmonary veins. And then the left atrium pumps blood through the mitral valve into left ventricle. Left ventricle pumps blood through the aortic valve into the aorta, and then it goes back out to the body. We usually think of this as 2 different circuits. The pulmonary circuit, which is the heart to the lungs, back to the heart, and the systemic circuit, which is the heart to the body, back to the heart. And while there's a left and a right side to the pulmonary circuit, there's an upper and lower, or superior and inferior component to the systemic circulation. And then also in the key up here, they have red and blue keys to show you which is which for arteries and veins, because it's different in pulmonary versus systemic circulation. That kind of shows you the whole layout of the system. I want to show you one more thing that's actually going to get into the next material, but I want to show it to you before you look at the heart models, so you can look at where that would be on the heart model too. And the next thing we're going to talk about is the cardiac conduction system, the cells that are specialized for the electrical signals that travel through the heart. And what I want to show you is 2 of the important parts of that cardiac conduction system are the SA node right here and the AV node right here. SA stands for sinoatrial. It's in the wall of the right atrium. And it's actually in a rather superior position, very close to the superior vena cava. The other one, the AV node, is the atrioventricular node. And again, AV, like AV valves, is referring to it being at the junction between the atrium and the ventricles. It's still in the atrium, actually. It's in the wall of the right atrium, but it's right next to where the ventricles are. So when you're looking at the models, what I'd like you to do, besides finding the 4 chambers, the 4 vessels, and the 4 valves, is also in the right atrium, look for where those things would be. They're not necessarily going to be labeled or anything, but think about the sinoatrial node is going to be up in the superior right atrium, and the AV node is going to be kind of just medial to the AV valve there. And I'm not quite positive. I don't have a key for this thing, but there's a little writing on the inside of that. That might be where those are located. I don't think it is, but it might be. So you can look at the heart models to basically see the 3-dimensional structure of the heart, find those 12 things, the 4 chambers, 4 vessels, 4 valves. And I want you to, as you're finding those 12 things, to think about where they fall in the process of the blood pumping through the heart. And traditionally, we start with the blood entering into the right atrium. Okay, so think about what vessel's carrying it into the right atrium and what valve is separating the right atrium from the right ventricle, and just follow the whole path through like that. Now, these 4 on this card are from next door. The 3 on that card are actually from this room, but somebody was using them this morning, so they have them on a card already. Between the 2 cards, there are 4 of these really big models, 2 of these medium-sized models, and then there's this 1 small model over here. Students will often try to avoid the big models because they're old and clunky, and I can sort of kind of survive. But they're actually somewhat the better models. What I really like about it, it's the only model that shows the heart in relation to other organs in the mediastinum. So there's an esophagus and a trachea in both of these that we find between the heart and the descending aorta, because that's where they're located. That's kind of nice. The wall parts to these big ones tend to fall off, so you might not see them attached, but they should be around. All of them should be here. And if you take it off, you can't get it back on. Don't worry about it too much. I do want to point out something for the big models. If you take the wall piece off that exposes the right ventricle, and you're looking for the pulmonary valve, you'll be disappointed. Pulmonary valve is actually part of the wall piece here. Why they did it that way, I'm not sure. But so you don't see the pulmonary valve in the heart here because it's come off in the wall there. But other than that, it's all pretty straightforward, I think. So again, what I want you to do is, using these models, follow through those 12 structures that I mentioned, which let me put that back up on the board as best I can. Okay. Okay. I want you to look at these. There we go. They all fit. In regards to the direction the blood is pumped through the heart. Also, look inside the right atrium and think about where those 2 nodes would be as I described them, just so you have some point of reference when we get to the next part there. So I want to give you time to look at these. I usually give about 15 minutes to do that. And then you're also going to take your normal break, 20 minutes to do that. So by the clock up there, I want you to be back in here by 1:45. If you'd like to take a break and then come in and look at the heart models, that's fine. If I look at the heart models and then take a break, that's fine. But at 1:45 by that clock, we're going to continue on and look at the next part of the stuff.